Thank you very much, Dr. Mohammed. Good afternoon, everyone. And thanks for the organizer for their kind invitation. So for the topic of today, I mean, it's very exciting times for the NTRK band treatments. And in my talk, I will focus mostly on the laboratory side. What is the best method to detect NTRK, when to use some of these methods, and the actual, going through the behind the scene will help us to understand when to use some of these methods.
Dr.
So
Saeed.
let me just open my slides.
Yeah, could you slide show your presentation? Yes, now it's good.
Okay.
Thank you.
Perfect. So the central dogma in genetics and biology is that DNA will give us RNA, RNA will give us proteins, and if we apply this concept to the NTRK genes, we have 3 genes, as everyone knows. NTRK1, for example, will give us 10 different RNA transcripts, and the eventual output would be a TRKA. 8 transcripts for NTRK2 and 21 transcripts for the 3rd gene. So it will be very essential idea here that will go with us through the slides to understand what we see here, because some of the tests will be used at the DNA levels, others will be at RNA, and some will be on the protein level. They will have very different characteristics. So what we are dealing here with is the gene fusions, and just for everyone to be on the same page, we are talking about a break part of a chromosome at a certain position and linking it to an abnormal position in a different chromosome, sometimes on the same chromosome. What you see here is a translocation between ETV6 and NTRK3 from 2 different chromosomes, 12 to 15. And the results is giving us abnormal linking between 2 parts of the gene, and finally this will give us the fusion gene with a different characteristics and function. Now this translocation, sometimes there's a lot of translocation that does not end up in RNA or proteins. Sometimes it will give us RNA only and does not translate into protein, so they may not have an important effect on the cancer. But in this case, the chimeric protein has a carcinogenic effect and lead to the progression of the cancer. And listing the number of partners with NTRK, we will find that there's at least 30 different genes that can be translocated with one of the 3 genes. In this example, you see there's at least 10 different genes that goes with NTRK1, another 8 or so with NTRK2, and probably 4 or 5 with NTRK3. So this could be a huge challenge for us in the lab, and different methods will be able to detect different kind of gene fusions. So what we are doing, why we are doing all of this? We are doing this because of the beautiful treatment that is available today, and I will not go into this. Dr. Fatma gave a great presentation and covered most of this. But this is the eventual goal for us, is to target a selection of therapies for the patients. So what are the toolbox that we have in the lab? If we look at the DNA level, we can use reverse transcriptase, real-time PCR, or FISH, or NGS. Similarly, on the RNA level, we can use the same methods. At the protein side, we can use immunochemistry. And I'm only listing here just these methods, but in reality, there is tens, if not hundreds of different methods, but these usually are the clinical methodologies that's used in standard of care pathology departments. So let's just quickly go through real-time PCR and see how it fits our needs for the NTRK testing. The concept with real-time PCR is very simple. We are interested in a very specific part. In this case, let's take ETV6 with NTRK3. So we design a small primers that will target this fusion. And through multiplication, it will give us 2 copies, then 4, then 8, until we reach millions of copies. Now, with naked eyes, we cannot see this. So what we end up doing here is we add a fluorescence that's attached to this target. And by using a laser, it will give us a signal that will measure the intensity of this fluorescence. And what we will end up is having a curve like this. The area of the curve, the higher the concentration of a target. And if we don't see a target, there is no curve. This means it's negative for that specific NTRK. Now, let's see what is the advantages of this method. First, it is extremely sensitive and very, very specific, and it's widely used in so many labs. This is one of the major basic methods, but there is a very big drawback for this method. The first thing is, for every specific gene fusion, we need to have primers, which means, like you saw, we have 30 or more fusions. We need to design 2 primers for each fusion. This is almost 60 different reactions if you want to cover the non-NTRK fusion. So this is almost impossible because the cost of each reaction, let's say it's 100 riyal, we are talking about probably 6,000 riyal for just one gene for the fusion, and it's also troublesome. There isn't enough DNA or RNA that can suit or be enough for the whole reaction. So from the get-go, real-time PCR is not going to be very helpful for the NTRK fusion. So let's move to another very commonly used method, the fluorescence in situ hybridization, the FISH. And the FISH in principle is similar without the copier. So we have a probe for NTRK gene, and we have in green color and red color for the ETF6. If these signals are close to each other, we know physically there is a fusion. And you can see in the small picture, the red dot and the green dot indicating to us that these 2 targets are close to each other, which tell us there is a fusion. And you can imagine the same problem will happen with the real-time PCR. For every fusions, we need to design a probe, and that's a lot of probes. So it's not cost effective. Luckily enough, there is a concept called break-apart probes. Basically, we don't care about the other partner B. We only care about NTRK3, NTRK2, NTRK1. So what we will do is we will put probes on both sides of the NTRK3. And if the signals, the green and the orange, always together, we know NTRK3 is intact and there is no breaking or translocation. It's only when these 2 signals is far away from each other that will give us the idea there is NTRK, there is a fusion with some unknown gene. So we know there is a fusion, but we don't know what is the partner. And that's very clever and good because we can detect novel fusions. We know there is a fusion, but we will never know what is the other partner using this method. So FISH is slightly a little bit better than real-time PCR, but we still need 3 sets of probes for NTRK1, 2, and 3. And FISH, for those who's working on cytogenetics, know it's not the easiest or most fun methods. There's a lot of technical issues. It's need a highly expert people to read the slides, and it's not always easy to score for the FISH results, although it is very sensitive and very specific. So we can use it, but only when we have to use it, if that makes sense. So moving to the next generation sequencing, which started almost a decade ago, but recently gaining a lot in the clinical labs in the last 5 years. And the idea is very simple, but it's so powerful. Next generation sequencing can read a small fragment of DNA or RNA. It has to be chopped by different enzymes or physical methods into small parts, usually 50 to 200 base pairs in length. And then there's many methods. One of them is called Illumina solutions. And basically they have a small glass and they put it on it millions upon millions of these small fragments of DNA. They use a laser and the fluorescence to read every base on the slide. And it will create an image like the one on the right side. So what you see here is dots that will be built sequentially on top of each other as the DNA is being synthesized. And this will be translated in a text file. This text file will contain millions or billions sometimes in every run of short segments. And here is representative of 5 different sequences. It's random, coming from different places in the genome. So in this format, it's absolutely not useful for us. We cannot read it. We don't know what it is. So what we need to do using bioinformatics is to realign or map it to the human reference genome. So you want to know where is this read located. So there will be a program that will take every read, scan the whole genome, and try to figure out where it is. Once it finds it, it will put it there and says this reads come from this location. And we will do this for all of the reads. So once the read mapping finishes, we go to the next level. It's good to know that it's mapped, but we don't know what are the mutations. So the second step is going to be annotation. Basically, we want to say, what is this location means, where does it come from? And you can see here, there is 2 different, for example, colors. One is coming from the NTRK, but the other part of these reads is coming from another gene. In this case, the ETF, for example, ETV6. And this is what will tell us in this location, we have evidence of multiple reads that span both ETV6 and NTRK, although they're not supposed to be normally part of the genome. You can see the red part here is not matching the reference genome highlighted in yellow color. So this is how NGS tells us about the translocation. The beauty here is it can do this for thousands of locations. So we can cover almost all NTRK partners in one reaction. It will tell us also exactly what is the location in the exon. It may not be important today, but as more data comes from these experiments, we may actually subclassify the patient based on the location of the translocation into prognostic values, or even different response to treatments, like we have seen already with the ALK fusions in lung cancer. So the key important question here is, should we sequence the DNA or should we sequence the RNA part? And that has a huge impact, and rarely people understand this because it's too technical, but I will simplify it here. So at the DNA level, this is a very quick 101 molecular biology for the gene structure. We have every gene composed of different components. The red part's called the exon, and this will translate it into protein. The light blue is the intronic parts. This is non-coding parts. And when DNA is being translated to the RNA, what we usually have is the messenger RNA. So only the exons, intron, will be shipped out and removed. So we end up having only the coding parts. And this has a huge effect on the NGS, because if we look at the NTRK genes and see what is the length at the DNA level, you will see that NTRK1 is almost 20,000 base pairs long. NTRK2 is 357,000, and similarly NTRK3. So collectively, we're talking about 775,000 base pairs. And every pair will need to be sequenced, which translates into a cost, which translates into fewer sample per run. So it's not cost effective to go with a DNA level. While if we just focus on the transcript or the RNA level, you will see that it represents much smaller part of the DNA. So we're really talking about 31,000 in total, which means it's a smaller version. It can be sequenced at a higher depth. We will have a better signal and a better detection at the RNA level, especially for these 3 genes. So ideally, and from theoretical perspective, RNA is the way to go, not from the DNA level. So what are the advantages of NGS? Extremely sensitive and very specific. We can multiplex it, not only NTRK. At National Guard, we have more than 50 genes that's being sequenced for all solid tumors, including NTRK 1, 2, and 3. So this gives the patients a much wider options for multiple genes, and we don't consume the tissue, which is always a concern. It can detect both novel known and novel fusions, unlike the real-time PCR. So this is a huge plus because we don't know all the partners yet with NTRK genes. We want to discover as we move forward, and the NGS is beautiful for that. So it serves both clinical and research goals.
5 more minutes.
Thank you. And for the RNA parts, it's very nice to have the RNA and the advantage compared to the DNA NGS, because RNA could actually put us closer to the protein. Not every fusion at the DNA level will be translated into RNA and RNA to protein. So sometimes you might find a fusion at the DNA level, but it does not have a protein, and the patient will never respond to treatment because they never actually have the fusion proteins. So when we sequence the RNA, it's put us closer to the actual dynamic of the biology of the tumor in that patient. So the last part is comparing very common and loved methods in the pathology, histopathology, specifically the immunohistochemistry. And immunohistochemistry is cutting to the chase. It just go to the protein directly. It doesn't waste any time with RNA or DNA. If the protein is there, let's pick it up. And they do have a trick immunohistochemistry. It's very beautiful. It's widely available, very low cost, and relatively easy to read. It will stain the slides with this brown color, showing there is a detectable fusion. The big problem here is that it's not very sensitive, nor it's very specific. And we will see some data showing the sensitivity and specificity of this method. But it's a beautiful method for screening, especially in cancer that does not have NTRK fusion, like colon frequently. So we cannot do NGS on all colon cancers, so it's better to use immunohistochemistry as a screening method, like Dr. Fatma mentioned previously. Caris Life Science published a beautiful paper a couple of years ago. They did 11,000 patients. They did NGS at the DNA level, RNA level. They did immunohistochemistry as well as BDL1. And from this cohort, they picked 31 positive cases with NTRK, which represent 0.27%, which is extremely low. But they compared the power of immunohistochemistry with NGS in detecting these NTRK fusions, and they found the sensitivity is 75% for the NTRK for the immunohistochemistry, which is not great, but it's not bad either. And it's situated the immunohistochemistry as a screening tool. So in summary, if we compare these 5 methods, immunohistochemistry, FISH, real-time PCR, NGS DNA, and NGS RNA, you will see the cost is increasing as we move toward the NGS. Turnaround time is also associated with NGS. Usually it will take 10 to 15 days, while immunohistochemistry and FISH probably 1 or 2 days. When we talk about novel fusions, it's mostly NGS that can detect the partners, but the others are not, especially immunohistochemistry and real-time PCR. When we talk about multiplicity or number of tests in one reaction, then NGS is the winner. We need only 1 test to pick all the partners, as well as the immunohistochemistry, not the case with real-time PCR or FISH. Sensitivity and specificity, I think it's very clear that RNA NGS is the best and the most sensitive methods. Immunohistochemistry is a medium to a little bit high. And the complexity also increasing from the top to the bottom. And finally, the subjectivity. So immunohistochemistry and FISH require subjective experts to give their opinion, while real-time PCR and NGS is data-driven and does not require a human intervention a lot for these fusions. I will end up with this algorithm for testing. It was published by the ISMO last year, and they are suggesting to detect NTRK fusion in histological type that is known to harbor NTRK is just to go for NGS directly or FISH. But if it is not known to have NTRK, like colon cancer, for example, then maybe if the NGS is part of the operation and you have money, then by all means. If not, then screen with immunohistochemistry and if it is positive, confirmed by NGS. That's what I have. Thank you very much for listening.